So the next topic is biosynthesis of glycosides. So in this biosynthesis of glycosides, generally the glycosides are synthesized in two parts. In the first part, the biosynthesis reaction it takes place with the uridyl triphosphate with sugar 1-phosphate to form UDP, so which in turn in the second step reacts with the A-glycone moiety. So let us see the first step. So uridyl triphosphate, UTP, is nothing but uridyl triphosphate. This reacts with the sugar 1-phosphate in presence of uridyl transferase. So here what happens is 1-phosphate is removed from UTP and the sugar molecule get attached to the uridyl diphosphate. UTP means uridyl triphosphate. When you remove 1-phosphate unit, it becomes UDP. Okay, so that phosphate unit it combines with the phosphate which is attached to the sugar. Okay, this sugar in turn attaches to the UDP. Okay, so in this reaction the UDP will be converted into UDP and the sugar molecule get attached to the UDP formed from UTP. Uridyl diphosphate sugar is formed, and this phosphate from UTP and this phosphate both will be eliminated in the form of pyrophosphate. Okay, so the first step involves the formation of UDP sugar, which is essential for the formation of glycosides in the second step. So this reaction is catalyzed by uridyl transferase enzyme. So second step, in the second step UDP sugar, that is uridyl diphosphate sugar, it combines with the A-glycone moiety. Okay, this A-glycone moiety will be synthesized from different pathways, various pathways like flavonoids, alkaloids, steroids. It depending upon the chemical nature, it follows either the shikimic acid pathway, acetate mevalonate pathway, acetate malonate pathway, and they involve in the reaction to form the glycosides. So here what happens is the sugar molecule get detached from here and this combines with the A-glycone moiety. So one hydroxy group from sugar and one hydrogen from A-glycone moiety will be eliminated in the form of water to form a glycoside, and the linkage formed between these two is called as glycosidal linkage. So here in the second step what happens is the sugar attached to the UDP, uridyl diphosphate, it combines with the A-glycone moiety to form the glycosides in the presence of glycosyl transferase enzyme, and this UDP will be converted to UTP, uridyl triphosphate. So this is the biosynthetic pathway of some of the flavonoidal A-glycones. Just now I told you. So depending upon the nature of or chemical nature of A-glycone moiety, they will be synthesized from various pathways. So here shikimic acid pathway is involved in the formation or synthesis of flavonoidal glycoside, A-glycone moiety. So from shikimic acid we know that we'll get corysimic acid, and from there we'll get anthranilic acid and prophenic acid. Prophenic acid involved in the synthesis of phenylalanine. Okay, so phenylalanine, it in turn it forms the cinnamic acid. So this cinnamic acid it combines with this acetate malonate pathway. So acetyl CoA and malonyl CoA will be involved, combined together and gives some intermediates, and that intermediates combine with the cinnamic acid to give C15 intermediate. So this C15 intermediate forms the cyanidin, and on the other hand it also forms quercetin. Okay, so this is about the synthesis of flavonoidal A-glycone moiety. So next, biosynthetic pathway of other flavonoidal A-glycones. So this also follows the shikimic acid pathway. So from shikimic acid pathway it gives phenylalanine, cinnamic acid, and from cinnamic acid vanillin is derived, and this cinnamic acid also helps in the formation of coumarins, arbutin, helicin and salicin. Okay, so this cinnamic acid, it undergoes reactions and forms O-coumaryl glycoside, which in turn gives the coumarins. And on the other hand, cinnamic acid, it also gives hydroquinones, and from the hydroquinones arbutin is formed. Okay, cinnamic acid, it also forms helicin, and this helicin gives you the salicin flavonoid. Okay, so next, in the same way, the biosynthesis of amidin and related compounds. Okay, so amidin and related compounds involves the involvement of 8 acetate units, which gives poly beta ketomethylene acid intermediate, from which we get amidin and other relative derivatives. And cyanogenetic glycosides are also synthesized from the shikimic acid pathway. So this shikimic acid pathway gives rise to phenylalanine tyrosine, and from valine and isoleucine we will get linamarin, lotastralin, and from tyrosine we get durin, and from phenylalanine we get pronacin. Okay, so these are the applications of biosynthetic pathways, various biosynthetic pathways. So the next pathway is amino acid pathway. So we know about the amino acids. In the name itself we can see amino and acids. That means the compound which contains both the amino group and acid group within it. Okay, the same compound, the compound, it contains both amino groups and acid groups. Okay, now we will see the biosynthesis of these amino acids. So amino acids are found in either free state, like we can see phenylalanine, tyrosine, tryptophan, valine, isoleucine, all these are the amino acids, right? So either they found as amino acid, that is free state, or as basic units of proteins. Amino acids are also the building blocks of proteins, right? So either they occur in the free state, or they also found as basic units of proteins. Okay, proteins are nothing but the polymer of amino acids. It contains more number of amino acids. So amino acid synthesis, it involves various pathways, that is intermediates from various pathways, that is glycolysis and TCA cycle, that is Krebs cycle, and also aromatic amino acids are synthesized via shikimic acid pathway, and it also involves pentose phosphate pathway. Intermediates from glycolysis, pentose phosphate pathway, and also Krebs cycle or TCA cycle. So these amino acids, generally they act as precursors from some secondary metabolites, like we have seen flavonoidal aglycone moieties, and some of the alkaloids are synthesized from this amino acids. So these amino acids, they act as precursors in the biosynthesis of secondary metabolites. So in general, the amino acids which occur in the nature are alpha amino acids. Okay, so next, so total we have 20 amino acids. So they are divided into essential and non-essential amino acids. Okay, so here you can see the general formula of amino acids. So general, if you see the general formula, see here. So on the same carbon we have the amino group and also carboxyl group. Okay, so amino acids, amino group and acid group. The compound which contains both the amino and acid groups are nothing but amino acids. Okay, so next, amino acids, generally we have 20 amino acids, among which 9 are essential amino acids. Essential amino acids means which cannot be synthesized by the body, so you have to provide it from the external diet. Okay, so we have 11 non-essential amino acids. Non-essential amino acids, there is no need to provide it from the external source, they are synthesized within the body. Okay, so here you can see the essential amino acids: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. Okay, so these are the non-essential. 11 non-essential amino acids and 9 essential amino acids are there. Okay, so as I already told you, the amino acid pathway starts from the glycolysis, and also it involves some of the amino acids are synthesized from the intermediates of Krebs cycle and alpha ketogluteric acid. So next, how the nitrogen enters into the metabolic reactions. So here the nitrogen enters the metabolic reactions by reductive amination. Okay, so undergoes reduction and amination will be taking place. If you see the alpha ketogluteric acid which undergoes reductive amination, that means amine group will be added to this ketogluteric acid and forms aliphatic amino acids. Here you can see this is alpha ketogluteric acid, so it contains a keto group and also 2 carboxylic groups in its structure. So this undergoes dehydration, reductive amination. Okay, so in presence of amine it undergoes reductive amination, so the amino group get attached onto the carbon containing keto group. Okay, so this happens in presence of the NADPH, okay, which will be converted to NAD. Okay, reduction taking place. So this reaction is catalyzed in presence by the enzyme glutamate dehydrogenase. Okay, so when you treat the alpha ketoglutarate, it undergoes reductive amination and the keto group present in the alpha ketoglutarate will be replaced with the amino group. So here you can see, so this undergoes amination and forms glutamate. Okay, so on the second carbon, so here we can see, here we have the keto group and here we got the glutamate, amino group is substituted. Okay, and this further in presence of ATP will be converted into ADP. So if you further go for the amination, so here it will be attached onto the carboxylic group. This happens in presence of glutamine synthetase. Okay, so it leads to the formation of glutamine. So this is called reductive amination. Okay, so what do you mean by transamination? Transamination means if you take 2 compounds, one containing keto group and one containing amino group. Okay, so here we are taking amino acids. Okay, so this is keto acid. Okay, so it contains acidic group and one keto group. Okay, so in order to differentiate, we marked the methyl group with the R1 and R2. Okay, so now when you treat this keto acid with the amino acid, so there is a transamination reaction in between these two. This reaction is catalyzed by transaminase enzyme. Okay, so in transamination reactions, what happens is the amino group which is present on one compound will attack this keto group and it will get attached to this group, and this keto group will be transferred to this R2 compound. Okay, so here we have R1, the amino group will be transferred here and this keto group will be transferred here to form new compounds. So here we can see, so this compound, it has R1 and this contains R2. Okay, there is a difference in the methyl group. Right, now this R1 containing compound, it has keto group here, right? So here the R2 containing compound, it has a amino group here. Okay, so in transamination, this compound will be shifted, transferred here, and this keto group, complete keto group, is shifted to this another compound. Okay, so now this will shift here, right? So here you can see this will shift here. Okay, so now what happens is the R1 containing compound, initially it had keto group. Now you see if you see the products, the R1 containing compound, it has the amino group. Okay, so now here we got amino group, whereas if you see R1, it has keto group. This is called transamination. Okay, so the R2 containing compound, it got keto group. Okay, so the transfer of amino groups takes place in transamination reactions. Okay, so here also we can see these are the examples for transamination reaction, because during the formation of amino acids, in the last step it undergoes amination, right? So this is important to know. What do you mean by this transamination? Okay, so here you can see alanine, it is an amino acid, it contains an amino group and an acid group. Now we are taking alpha ketoglutaric acid. Okay, so this alpha ketoglutaric acid, it contains a keto group in its structure. Okay, right. So when you take an amino acid, it undergoes transamination. Okay, this reaction is catalyzed by glutamate pyruvate transaminase. Okay, all these transaminase reactions are reversible in nature. Okay, so in previous slide we have seen the general example of transamination. Okay, so these two products can undergo transamination and give this amine and keto groups, right? So this again undergo reversible reaction and give you the former ketone and amino groups. Okay, the same way here we are taking one amino acid and one alpha ketoglutarate, right? So this under glutamate pyruvate transaminase enzyme, the alanine, it contains amino group, right? So this is converted to keto acid because as we already discussed, see here the amino group is present on the alanine. Okay, so the keto group is present on the alpha ketoglutarate. Now this is transferred here and this keto group is shifted so this keto group shifts onto the other group, right? Here you can see this shifts here. Okay, the total amino group will be shifted here and the keto group will be shifted here. So here now you can see the products, right? So here in place of amino group we got the keto group. Okay, so in place of keto group we got amino group. Now the valine is the amino acid here. Now one more new amino acid is formed in case of this products, right? So here we got the pyruvate, alanine got converted to pyruvate and the alpha ketoglutarate is converted to glutamate. Amination is taking place in place of keto group in the alpha ketoglutarate. So this can be reversible also. Again the pyruvate can be converted to alanine and the glutamate can be converted into alpha ketoglutarate. So one more example, you can see aspartate contains amino group, whereas the alpha ketoglutarate, it contains the keto group. Okay, so in this also the same reactions takes place. So this keto group shifts here and the amino group replaces this keto group. Okay, so this reaction is catalyzed by glutamate oxaloacetate transaminase. So here you can see the aspartate, it contains the amino group. Now here we got the keto group. This is called transamination, right? The amino group is replaced by keto group and the keto group is replaced by the amino group. Okay, so the aspartate is converted to oxaloacetate and alpha ketoglutarate is converted into glutamate. Okay, so this is known as transamination. Okay, right. So this reaction is important in amino acid pathways. Okay, so next we will see the amino acid pathway. So this is the general amino acid pathway, right? So here it involves the glycolysis. Okay, so it starts with the glycolysis, right? So here you can see the glucose is converted to glucose 6-phosphate G6P, right? And so this undergoes reaction and forms intermediates and is converted to 3-phosphoglycerate, 3PG. Okay, 3-phosphoglycerate, and from here again we get phosphoenol pyruvate. Okay, so the blue color it indicates the glycolysis, and yellow color these products are obtained from the pentose phosphate pathway, and these are obtained from the intermediates obtained from Krebs cycle or TCA cycle or citric acid cycle. Okay, so these three compounds, these three are the intermediates derived from glycolysis, and these two intermediates derived from pentose phosphate pathway, and these two intermediates are derived from Krebs cycle, which involve in the formation of amino acids. Now this is glycolysis. So glucose 6-phosphate is converted to 3-phosphoglycerate, and this gives phosphoenol pyruvate. So this we have already discussed. It is a precursor or starting material in the shikimic acid pathway. Erythrose 4-phosphate, it also involved in the shikimic acid pathway. These two are the precursors of shikimic acid pathway. So we have already seen from shikimic acid pathway we get 3 aromatic amino acids, that is phenylalanine, tyrosine and tryptophan, right? So erythrose 4-phosphate is obtained from the pentose phosphate pathway, and phosphoenol pyruvate is derived from the glycolysis, and these two involve in the shikimic acid pathway, and they finally give the corysimic acid, and the corysimic acid in turn gives prefinic acid and anthranylic acid, and the prefinic acid it gives phenylalanine and tyrosine, and the anthranylic acid it gives tryptophan. Okay, so from the shikimic acid pathway we get phenylalanine, tyrosine and tryptophan. Okay, so now here you can see from glucose 6-phosphate, so it is also forming some intermediates, and finally it gives ribose 5-phosphate, which gives histidine. Okay, so from 3-phosphoglycerate also we are getting serine. Serine is also an amino acid. And from pyruvate also it forms alanine, valine, isoleucine and leucine. Okay, and next this pyruvate, so it undergoes amination and forms alanine, and this pyruvate also forms some intermediate and finally forms alpha keto isovaleric acid. So this alpha keto isovaleric acid, it in turn forms the valine, and it also forms alpha keto isocaproic acid. From alpha keto isovaleric acid we get alpha keto isocaproic acid, which undergoes amination and forms leucine. Okay, and it also forms alanine by amination. Okay, so valine from alpha keto isovaleric acid and leucine from alpha keto isocaproic acid, and this pyruvate undergoes amination and forms valine. So, next, this pyruvate also involved in it gives acetyl CoA, and from acetyl CoA we get citrate. Okay, so this citrate undergoes reactions and some intermediates will be formed, and finally it gives alpha ketoglutarate. So here we are stressing upon the intermediate which involve in the synthesis of amino acids, right? So in the whole citric acid cycle or Krebs cycle, these two intermediates, alpha ketoglutarate and oxaloacetate, these two involve in the synthesis of amino acids. Okay, alpha ketoglutarate, this undergoes reductive amination and forms glutamate, and from glutamate we get proline, and this in turn it gives glutamine and also ornithine. From ornithine we get arginine. Okay, and we also have one more intermediate of this citric acid cycle, that is oxaloacetate. Oxaloacetate, it undergoes amination and forms aspartate. Okay, this is aspartate. So this in turn it forms homoserine, and from homoserine we get threonine, isoleucine and also cysteine, methionine. Okay, so here homoserine will be formed. From homoserine we get threonine, lysine, methionine and cysteine. Okay, isoleucine is also formed. Next, from shikimic acid pathway we get phenylalanine, tyrosine and tryptophan. So this is about the amino acid pathway. Okay, so in amino acid pathway, so generally in amino acid pathway it involves the intermediate from glycolysis, pentose phosphate pathway and citric acid cycle. Okay, so glycolysis, it gives glucose 6 phosphate, which in turn gives ribose 5 phosphate, and finally it gives histidine. And from 3-phosphoglycerate we get serine, then undergoes amination and gives serine. And from phosphoenol pyruvate, so this phosphoenol pyruvate is an intermediate of glycolysis. This acts as a precursor for shikimic acid pathway. And erythrose 4-phosphate from glycolysis, this also involves in the shikimic acid pathway. These two involve in the shikimic acid pathway and helps in the synthesis of phenylalanine, tyrosine and tryptophan. So this phosphoenol pyruvate, on the other hand, it gives pyruvate. So this pyruvate gives acetyl CoA, and from acetyl CoA we get citrate. So this is our Krebs cycle. So before going to the Krebs cycle, the pyruvate, it involved in the synthesis of alanine, valine, isoleucine and leucine. This undergoes amination and gives valine, alanine, isoleucine and leucine. So on the other hand, it also forms acetyl CoA, and acetyl CoA it gives citrate. So this leads to the TCA cycle. So in the TCA cycle, these two intermediates are important, alpha ketoglutarate and oxaloacetate. So from alpha ketoglutarate we get the amino acids such as glutamine, proline, arginine, whereas from oxaloacetate we get asparagine, methionine, threonine and lysine. So this is about the amino acid pathway. Okay, so by this we complete all the 4 pathways, that is shikimic acid pathway, acetate pathway. As in acetate pathway also we have 2, acetate malonate pathway, acetate mevalonate pathway and amino acid pathway.